Welcome back, folks. This is 2-3 to 2-5. This is the basic energy concepts. This is not going to be hard thermodynamics. This is going to be just some basic concepts that we deal with in anatomy and physiology. Eventually in your life, I hope you take a thermodynamics course. It will depend on what your goals are. Probably if you're going to medical school, you'll have to. If you're going to PA school, probably the extent of your thermodynamics will be your chemistry class. So, and nursing school, same, it'll be chemistry class. So here we go. First of all, we have three different types of systems. We have an isolated system, and what that means is you don't exchange, you exchange neither energy nor matter with the universe, the surroundings. By the way, I'll call the surroundings the universe, or maybe I'll just call them surroundings, keep it simple. But if you hear someone talk about a system in the universe is the surroundings, and the system is whatever you're looking at. And the system could be the cell, or the system could be planet Earth. So these are just terms we use to talk about the exchange of energy and matter. So a closed system, nothing's exchanged between the system and the universe. That means energy's not exchanged and matter's not exchanged. That's an isolated system. Probably doesn't exist. Well, it probably does exist. Maybe and I'm not really sure, I'm speculating, but maybe in a galaxy, maybe a galaxy is an isolated system. I don't know if a galaxy exchanges energy and matter with another galaxy or not. I really don't know. But relative to biology, an isolated system doesn't exist. Okay. Then we have closed systems. Closed systems exchange energy with the universe, but not matter. So there's no matter exchange with the universe. Earth would be a closed system. We exchange energy with the universe, but we don't exchange matter with the universe. It's not like Earth gives matter to the moon, but energy definitely is exchanged between Earth and the universe. So that would be a closed system. And then we have an open system, and in an open system, matter and energy is exchanged freely with the universe. So that's an open system. Of course, cells are open systems. I'll say that again. Cells are open systems. You need a constant input of energy, and you need a constant input of matter, and matter is given off as waste products, and energy is given off as heat. So cells are open systems. Okay. So we're going to always be talking about an open system when we talk about thermodynamics. What happens in what thermodynamics says is this. There's several laws of thermodynamics. First of all, the first law says energy can neither be created nor destroyed. You can change its form, but you can't create it or destroy it. So first law of thermodynamics, energy can neither be created nor destroyed. It can be transferred or transformed, but not created or destroyed. That's the first law of thermodynamics. What's the picture is showing you here is that the chemical energy in the deer that the cheetah is eating will ultimately be converted to heat when the cheetah runs. So the cheetah is going to eat the deer. It's going to make a molecule of ATP when it metabolizes the food source. Not just one molecule of ATP, moles and moles of ATP. And then when it runs and you hydrolyze that ATP, heat will be given off. So you are going to change chemical energy to heat energy eventually. So you can change energy. You just can't create it or destroy it. The second law of thermodynamics is the entropy law. With any transfer or transformation of energy, no transformation is 100% efficient. You're going to increase entropy of the universe with any transformation. All right, so that's the second law of thermodynamics. You will increase the entropy of the universe with every energy transformation or transfer. Now remember, that's entropy of the universe. Because what you're going to eventually say to me is, how can I say this when I eat a meal and I build muscles? Now, a muscle, when I build muscles and I add more proteins to them, they're very organized, and that's low entropy. And I'm going to say to you, I do make muscle cells with low entropy at the expense of the universe. The universe has higher entropy because I'm keeping my body together. In order for me to keep my arms in place and my legs in place and my eyes in their eye sockets, which is very low entropy, it's very organized and low entropy, I do that at the expense of higher entropy in the universe. And eventually, after we die, we will quit keeping our bodies together and we will degrade and decompose and become CO2 and water, and our bodies will have high entropy. Because I think you would agree that if your body is completely a lot of CO2 and a lot of water, that's a higher entropy than when your arms are in place and your eyes are in place and your ears are in place and everything's in place. So entropy is a statement of order or disorder. High entropy is disorder and low entropy is order. So you keep your bodies in place and together and organized at the expense of increasing disorder of the universe. You selfish human being, you. So that's the second law of thermodynamics. Whenever you graph an energy change, you would look at the energy of the reactants. Well, let me stop there. Let me go backwards first. In any reaction, any energy reaction, you have reactants becoming products. Reactants are sometimes called substrates, and you'll see why in a second, because nearly every chemical reaction that occurs inside a cell is catalyzed by an enzyme. Nearly every single one. So sometimes we call these reactant substrates. Now, it could be that you have to put energy into this reaction. It could be that you have to put energy into it. Or it could be that when the reactants become the products, energy comes out of that reaction. That could be the case. So you could put energy in, or you could get energy out. Now what happens is you look at the energy of the reactants. Let's just give this a number. Let's call it 10. 10 what? 10 calories, 10 joules, whatever you want to call it. That energy right there is an arbitrary number I'm calling 10. And then let's give the products an energy. Let's call that, I don't know, 3? Looks like a 3 if that's a 10. Well, what happened? Well, what happened is, in between going from reactants to products, I lost 7 units of energy. 7 units of energy came off. From 10 to 3 is 7. All right, so I lost 7 units of energy going from reactants to products. This is called an exergonic, or chemists call it, just so you know what they call it an exothermic reaction. The reason why chemists call it exothermic is because this energy is heat. That 7 units of energy is heat, so they say exothermic. Biologists call it exergonic because it's not always heat. Sometimes we couple this release of energy with a storage form of something else, or we couple it with something, so it's not always heat. All right, so we call it exergonic. When you have an exergonic reaction, which means it releases energy in the reaction, it's a spontaneous reaction. I'm not saying it's fast. I'm saying it's spontaneous. Please don't confuse the two. Saying something is spontaneous means it's going to happen by itself no matter what. But that doesn't say it's going to happen today. It might take 10 years. So I'm not talking about kinetics. I'm talking about thermodynamics. Exergonic reactions are spontaneous. They may be spontaneous and fast. They may be spontaneous and slow. When you have to add energy to the system, like you're doing here, system or reaction, whatever you want to call it, when I'm adding energy to it, it's called an endergonic reaction. And if this is 3, the reactants are 3, and the products are at 10, I must have added 7 units of energy to that reaction. I had to. To get from 3 to 10, you have to add 7. So this is an endergonic reaction, and it required energy to be driven forward. This is a non-spontaneous reaction. If you don't add energy, it doesn't occur. Okay. So that's some basic energy concepts there. Here's some more basic energy concepts. We can talk about energy of position, or energy of concentration, or chemical energy. This is showing you an energy of position. It's called potential energy. When you're way up here on the diving board, you have a lot of potential energy. What's going to happen is when you jump off the diving board, that potential energy or energy of position is going to be converted to this kinetic energy or energy of motion. So this is kinetic, and this is potential. When you jump off the diving board and get down into, say, the pool or the lake, you have less potential energy because you expended some of your potential energy as kinetic energy. By the way, when you hit the water, you made a splash and made some noise, and that's energy as well. This picture right here is showing you concentration energy of a concentration gradient. What happens is I have a lot of these solutes all in one area, and they're all vibrating. Every molecule vibrates unless you're at absolute zero. By the way, absolute zero, 0 Kelvin equals, I hope I can remember this correctly, negative 200 and it's either 73 or 93. Let me take a guess. Negative 200 and wow, I don't remember. I think it's negative 273.15 degrees Celsius. I could be wrong on that. You guys can look that up. Make sure that I didn't misspeak on this. 0 Kelvin is absolute zero. That's when molecules have no vibrational motion, no molecular motion whatsoever. And that's about negative 273.15 degrees Celsius. It could be a 9, I forget. Okay? So there you go with, we're never at absolute zero. So even on the coldest day in January when it's negative 40 degrees Celsius, which by the way is also negative 40 degrees Fahrenheit, even on the coldest day in January, our molecules have molecular motion, vibrational motion, because these are vibrating, they're bouncing off each other, and they spread out. And this is actually called diffusion. This is an exergonic spontaneous reaction. The only time this would not occur is when you're at absolute zero. Otherwise, this is going to occur. And then chemical reactions, they make and break chemical bonds. It always takes energy to break a bond. Takes energy to break bonds. Requires energy to break bonds. Releases energy when you make bonds. Release energy when you make bonds. Now, sometimes biologists, they don't forget that, but they use the nomenclature fast and loose. And I'll give you an example when I get there, and then I'll explain the truth to you. But sometimes we kind of ignore this basic principle. It takes energy to break bonds every single time, no exception. You release energy when you make bonds every single time, no exception. And I'll come back to why sometimes that's confusing later. This is showing you potential energy becoming kinetic energy, moving the wheel till you get to a more stable. Oh, by the way, when you have lower potential energy, you are more stable. Now think about that. If you're on the top of the Empire State Building, you have higher potential energy because you could fall off the Empire State Building. When you're down on the pavement down below, you have lower potential energy. When you're up on top of the Empire State Building, you are more unstable. When you're down on the pavement, you are more stable. Lower potential energy equals higher stability. And we talk about this molecularly too. We say that CO2 in water is more stable than C6H12O6. Okay? There's a lot of chemical energy in these chemical bonds. There's less chemical energy in these chemical bonds. These two are more stable than this one. You can burn sugar, C6H12O6. You can't burn CO2. So C6H12O6 is less stable than CO2 in water. All right, so that's showing you some energy concepts there. Oh, we have different types of chemical reactions. Okay, we're going to start talking about dehydration synthesis and hydrolysis. We'll talk about these chemical reactions. So we have different types of chemical reactions. By the way, this is the one that I was talking about before. I told you that ground state sodium is very reactive, can even be explosive sometimes. And we store this in the chemistry lab under either, I think it's probably in glycerol, or maybe it's an oil. I forget now. I don't know if they'd store it in glycerol. That's an alcohol. That might be reactive. I'd have to ask what they store it in. But ground state sodium is very reactive. Burns underwater, in fact. It's very unstable. But sodium in the cationic state is very stable. And it combines with chloride in the anionic state to make table salt. Well, look at this. Reactive sodium metal plus chlorine gas gives you table salt. This is a type of chemical reaction. All right, so the two main types of chemical reactions we're going to talk about are decomposition or hydrolysis reactions right here. Hydrolysis stands for hydrolysis, water lysis. You use water to break bonds. These are catabolic, which means they break down, and they're exergonic. Catabolic reactions are typically exergonic. And then we have synthesis or dehydration synthesis reactions, and these are build-up reactions. These build up, and these are anabolic and endergonic. Anabolic reactions are endergonic, they require energy. Catabolic reactions are exergonic, they release energy. So let's look at this. Let's look at the formation and the breakdown of some molecules. This is glucose and fructose becoming sucrose. Sucrose is a dimer. There's two monomers. Glucose is a monomer and fructose is a monomer. They're both sugar molecules. Put these two monomers together and you get a dimer. It's table salt, sucrose. Well, this is dehydration synthesis. Why is it dehydration synthesis? Because this H and that OH came out of the reaction as water. So in essence, I dehydrated these two monomer residues to make my dimer. I took water out of them. That's dehydration synthesis. That's anabolic and that's endergonic. Requires energy. Whereas breaking down the sucrose, hydrolysis, you use water to split the bond. This is catabolic and exergonic. Okay. I have something I should say to you at this point. I told you before, it always takes energy to break a bond, and it does. It always takes energy to break a bond. So how can I tell you that this is exergonic? Well, I'm breaking and making bonds. Am I not making an OH bond right here? I am. Now I'm breaking this glycosidic bond. I'm breaking it. It requires energy to break that bond. But I'm also making a bond. I'm making this OH bond right here. And I'm making this bond right here. So I'm making and breaking bonds. So how do you determine if it's endergonic or exergonic? You add up all the energy it required to break the bonds and all the energy it was released. Let me just make sure I'm saying this correctly. I might be misspeaking. You add up all the energy required to break bonds, and you add up all the energy released when you make bonds, and you see who's bigger. If making bonds releases more energy than it required to break bonds, then it's exergonic, and then vice versa. Okay, very, very important picture, not in your book. These catabolic reactions that release energy are coupled to these anabolic reactions that require energy. Now, the major universal currency of energy coupling molecule is ATP. In other words, when I run my catabolic reactions and I break big molecules down to small molecules, what are the small molecules? CO2 and water. I store the release of that energy as ATP. And then when I need energy to run my anabolic reactions, like building big muscles, I use ATP. So ATP is this universal currency of energy that couples my catabolic reactions, the energy released from my catabolic reactions, with the energy required for my anabolic reactions. Another way of looking at it, it's showing you that ATP is this coupling molecule. ATP is made from catabolic reaction and it's consumed in anabolic reactions. These other molecules here are electron carriers. These guys, NAD, NADP, and FAD, are electron carriers. Here's what they look like when they're carrying electrons: NADH, NADPH, and FADH2. These are electron carriers, and NAD and NADH are universal electron carriers. From the lowliest bacteria to humans, they all use NAD and NADH. FAD and FADH and NADP and NADPH are quite ubiquitous, but I'm not sure they're universal. But they are found across all the kingdoms and domains. All right, redox reactions. Redox stands for oxidation reduction, or reduction oxidation, however you want to say it. Before I run the video for you, oxidation is losing electrons and reduction is gaining electrons. All right, so oxidation is losing electrons and reduction is gaining electrons.
Displacement reactions are among the most common redox reactions. For example, a metal in a compound can be displaced by another metal in the uncombined state. When metallic zinc is added to a solution containing copper-2 sulfate, it displaces copper-2 plus ions from the solution. Zinc reduces copper-2 plus ions by donating 2 electrons to it, producing copper metal and zinc 2 plus ions in solution. Note that copper metal is depositing on the zinc bar. Also, during the reaction, the solution loses the blue color that characterizes the presence of hydrated copper-2 plus ions. Let's zoom in to view the reaction on a molecular level. Zinc atoms enter the solution as zinc 2 plus ions, losing 2 electrons. Zinc is oxidized. The electrons are gained by copper 2 plus ions, which are converted to copper metal. Copper 2 plus ions are reduced. Similarly, metallic copper displaces silver ions from a solution containing silver nitrate, AgNO3. Each copper atom reduces 2 silver plus ions by donating 2 electrons. Note the silver metal is depositing on the copper wire. Also, during the reaction, the solution turns blue due to the presence of hydrated copper 2 plus ions. Let's zoom in to view the reaction on a molecular level. Copper atoms enter the solution as copper 2 plus ions, losing 2 electrons. Copper is oxidized. The electrons are gained by silver plus ions, which are converted to silver metal. Silver plus ions are reduced.
Okay, so that's a redox reaction, oxidation reduction. Whenever one thing is oxidized, meaning it's losing electrons, something else must be reduced, which means it's gaining electrons. If you only look at the oxidation part of the overall reaction, it's called a half reaction. So I could just look at the copper part and say copper's oxidized. But that's only the half. That's only a half reaction. I then also look at the other half reaction, which is the reduction of silver, silver cation, I should say. Well, what does redox have to do with biology? Well, in biology, the electron carriers NAD plus and NADH are involved in redox reactions. NAD plus is in the oxidized state, meaning it can be reduced to NADH. NADH is in the reduced state, which means it can be oxidized to NAD+. Let's look at that.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, The products separate from the enzyme, and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
Okay, so biological redox, not every single time, but many times involves the electron carrier NAD plus, which can be reduced to NADH. And by the way, let me tell you what NAD, FAD, and NADP stand for. NAD stands for nicotinamide adenine dinucleotide. You'll eventually have to know that. The B vitamin niacin. The reason why you have to eat niacin in your breakfast cereal, when you read your cereal box, you'll see niacin. Niacin is used in the human body to make NAD. You need niacin in your diet to make NAD. FAD stands for flavin adenine dinucleotide. Flavin adenine dinucleotide, and the vitamin riboflavin is needed to make FAD. And NADP is just NAD with a phosphate group on it. It's all that's different is there's a phosphate group on NADP. So it's nicotinamide adenine dinucleotide phosphate. That's all. All right, so that's biological redox. Now, later, another mini lecture has more about enzymes when I do the proteins, but just a quick introduction to enzymes in this energy concept here. Enzymes are responsible for nearly every chemical reaction in a cell. I can only think of 2 or 3 reactions that occur in a cell that don't require enzymes. Enzymes do not change the thermodynamics of a reaction. Let me be clear. Enzymes do not change the thermodynamics of a reaction. If a reaction is endergonic without an enzyme, it's endergonic with an enzyme. If a reaction is exergonic without an enzyme, it's exergonic with an enzyme. All right, so if enzymes don't change the thermodynamics, what do? They change the kinetics. They speed up reactions. And I'm not talking about doubling the speed or tripling the speed. Some enzymes speed up reactions 10 to the 17 times. I don't even know what 10 to the 17 is. 10 to the 12th is a trillion. 10 to the 15th is a quadrillion. So 100 quadrillion. I do know what it is. Some enzymes speed up reactions 100 quadrillion times faster. And that's some of the better enzymes. A typical enzyme could make a reaction that might take 3 years happen in 1 second. And that's quite a bit faster. So enzymes change the kinetics of reactions. They don't change the thermodynamics. Now, how do they change the kinetics? They lower the activation energy. Now look at this dotted line right here. That's called the activation energy, or E sub A, of a reaction without an enzyme. So these reactants, now this reaction, by the way, is exergonic. If this amount of energy right here is 10, and this amount of energy right here is 7, then 3 units of energy came off. Exergonic, 3 units of energy. And this is a spontaneous reaction. But I'm not telling you how fast it is. It might take 3 years to occur without an enzyme. But when you add an enzyme, it speeds it up greatly. And now it only takes, say, 3 seconds or 0.3 seconds or even faster than that. So enzymes speed up reactions, and they do it by lowering the activation energy. With an enzyme, the activation energy is very low. Without the enzyme, the activation could be very high. So enzymes speed up reactions by lowering the activation energy. This is showing you an endergonic reaction right here, and this is showing you an exergonic reaction right here. By the way, this is showing you an exergonic reaction right here. So you're seeing 2 exergonic reactions and 1 endergonic reaction. All right. Let's look at a little video of
When
activation energy.
molecules collide, there must be a minimum collision energy for the reaction to occur. Lacking this energy, the molecules remain intact and no change results from the collision. The colliding molecules must have a total kinetic energy equal to or greater than the activation energy, which is the minimum amount of energy required to initiate a chemical reaction. Click on endothermic or exothermic reaction to view an animation of the energy profiles for each of these reactions.
Before I click on them, let me remind you that in biology, exothermic reactions are called exergonic, and endothermic reactions are called endergonic.
This is an energy profile for nitrogen dioxide, NO2, reacting with carbon monoxide, CO, producing nitrogen monoxide, NO, and carbon dioxide, CO2. Click on each of the regions, A, B, C, or D, as they appear to view the molecular changes that occur during each stage of this reaction. As the two molecules approach each other, an attractive force begins to form between one of the oxygens of nitrogen dioxide and the carbon of carbon monoxide, which is indicated by the dashed line. The bond between the nitrogen and oxygen of NO2 begins to weaken, and the bond length increases. When molecules collide, they form an activated complex, which is a temporary species formed by the reactant molecules as a result of the collision before they form product. In this reaction between nitrogen dioxide and carbon monoxide, the activated complex is NO2CO. The activation energy, which is the minimum energy needed to initiate this reaction, is 132 kilojoules per mole. At this point in the reaction, the bond between carbon and oxygen continues to strengthen, and the bond between nitrogen and oxygen continues to weaken. A bond is now formed between carbon and oxygen. The bond between nitrogen and oxygen has broken. The products of this reaction are nitrogen monoxide and carbon dioxide. The products are more stable than the reactants. The products have a lower potential energy. In this type of reaction, heat will be released by the reaction to the surroundings. The heat released by the reaction is 226 kilojoules per mole. This is an exothermic reaction. This is an energy profile for nitrogen monoxide, NO, reacting with chlorine molecules, Cl2, to produce nitrosyl chloride, NOCl, and chlorine atoms. Click on each of the regions, A, B, C, or D, as they appear to view the molecular changes that occur during each stage of this reaction. As the two molecules approach each other, an attractive force begins to form between the nitrogen of nitrogen monoxide and one of the chlorines of the chlorine molecules. This attraction is indicated by the dashed line. The bond between the chlorines begins to weaken, and the bond length increases. When molecules collide, they form an activated complex, a temporary species formed by the reactant molecules as a result of the collision before they form the product. In this reaction between NO and Cl2, the activated complex is nitrile chloride, NoCl2. The activation energy, which is the minimum energy needed to initiate this reaction, is 85 kilojoules per mole. At this point in the reaction, the bond between nitrogen and chlorine continues to strengthen, and the bond between the two chlorines continues to weaken. A bond has now formed between nitrogen and chlorine. The bond between the two chlorines has broken. The products of this reaction are nitrosyl chloride, NoCl, and chlorine atoms. The products are less stable than the reactants. The products have a higher potential energy. In this type of reaction, heat will be absorbed from the surroundings by the reacting mixture. The heat absorbed, or delta H, is 83 kilojoules per mole. This is an endothermic reaction.
All right. You have an overview of enzymes. We're going to do enzymes again in the next mini lecture. But before we go on, I want to point a couple things out. Way back in chapter 1, I say way back, it's only chapter 2, but we talked about schnapps, the most common elements in the human body, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. We talked a little bit about these trace elements. What happens is this: quite often enzymes are for the most part proteins. I'm lying to you a little bit, but for our purposes in anatomy and physiology, I think they're always proteins. If you take genetics from me, you'll learn about some enzymes that are actually made of RNA, but for our purposes, enzymes are proteins. Sometimes these enzymes, the protein portion is non-functioning, but it needs a and the reason being is because it needs a cofactor to be functioning. And a lot of these trace elements and even some of these other compounds will combine with enzymes and make them fully functional. So enzymes might need cofactors to function.